Good morning. I'd like to start by thanking the organizers of this seminar series, which I always find really enjoyable and I think is very important to our community. I'm really pleased to have been included. Thank you all for tuning in. I have recorded this session, so that recording is what you're watching right now. But I'm also present, and I've done that so that I can answer questions in the chat as the recording plays out. At the end of the recording, I'll come on live and I'll take your questions about the talk. So my lab is interested in sensory processing and in brain-wide sensory processing networks. And in addressing these questions, we have to think about the methods that neuroscientists have traditionally used to address sensory processing and indeed almost any process within the brain. Traditionally, there have been approaches where you can either see the entire brain, this would be like fMRI or EEG, you can observe activity across the entire brain, but these methods don't resolve individual neurons, and they don't permit you to see what individual neurons are doing as you observe activity across the brain. At the other end of the spectrum, you have electrophysiology, which allows you to see exactly what traditionally one neuron is doing, and now there are technologies that allow people to observe activity across hundreds or even a few thousand neurons. But at that end of the spectrum, you don't have the ability to see the tens, hundreds, of thousands, the millions of neurons composing local circuits, but also brain-wide networks. So historically, there's been this gap in neuroscience's ability to address those scales, those of circuits within brain regions and those of networks across brain regions, while resolving individual neurons that compose those circuits and those networks. So we're using a set of technologies in the lab that quite a lot of labs are using now to try to address those scales. So we and many others have adopted the use of genetically encoded calcium indicators like GCAMP. So these are proteins that bind calcium. They increase their fluorescence when they're bound to calcium. And the effect of this is that activated cells with elevated calcium can become brighter at the times when they're active. The effect of this, if you express a calcium indicator across the entire brain, is you have a light field of neurons with fluorescent signals, and it's arranged such that neurons, when they're active, blink. That then gives you, at least in principle, the ability to see the whole brain at cellular resolution. Now, there are two keys to being able to do this. One is the fluorescence microscopy method that you use to observe the activity. And the one that I'll be talking about today is light sheet imaging, or selective plane illumination microscopy. This is a method that I think many of you will be familiar with, where the excitation light, the blue light in the case of one photon imaging of GCAMP, the blue light is sculpted into a sheet that goes through the specimen. And then here, orthogonal to and parfocal with that sheet, you have an imaging objective that observes activity across that entire plane of the brain very quickly. This just goes up to a CMOS camera. You can then do volumetric imaging through a number of different methods that allow you to scan that sheet through the brain and look at different depths in close succession. So light sheet microscopy provides one of the key elements that you need to be able to see volumetric imaging throughout the brain, and the other relies on the optical properties of your model system itself. And this is where the attributes of Zebrafish come into their own. This is why our lab uses this model system. So this is a developmental time series. This is just a time-lapse movie of a developing zebrafish embryo. And what you're seeing here is a line that has a transgenic marker in motor neurons. You're looking at the spinal cord here, and you're looking at motor neurons in the spinal cord as they project axons out to the musculature that they will eventually innervate. And this movie highlights a couple things. One is that zebrafish are fertilized externally, and so you can observe their development from the time the egg is fertilized on without interfering with that development. And the other is that at early life stages, they're optically transparent. So you can see internal structures like the nervous system as it develops, or using GCAMP as it functions. You combine that with a brain that you can fit within the view of a 20x objective and within the depth range of a light sheet microscope. And you finally have the opportunity to do what I described in the first place, brain-wide imaging of activity at cellular resolution. So in this talk, I'll start out by talking about the particular design of microscope that we've done in our lab. We build our own microscopes for reasons that should become clear as I give the talk. I'll then talk a little bit about how we've looked at processing across multiple modalities using this approach, and then I'll focus on the example of the auditory system and a description we've recently done in the auditory system, before finishing by talking about the prospects of this approach for modeling human psychiatric conditions such as autism, specifically as they relate to subcortical sensory processing. So let's start by talking about the microscope and the design of the microscope that gives us what we need in order to look at these sensory processing networks. In order to get a zebrafish under a light sheet microscope, we first need a chamber to put it in. It's an aquatic organism, of course. So we start with a 3D printed chamber. We glue sidewalls that are just cover slips to this chamber, place the animal in agarose so that it will remain stable, place it on a platform within the 3D printed chamber, and then fill that platform with the water media that the fish live in. You can see the example of such a chamber here that now has its cover slip sidewalls that's now full of water. There is a fish in there, although you can't see it. And you can see here the optical elements, the objectives that are producing the light sheets that go into this animal's brain. And whenever possible, we use two light sheets that mesh at 90-degree angles, because that just provides us with a more uniform light field. So that's how we're illuminating a plane within the brain. And this is that imaging objective that I referred to in the previous slide. This is parfocal width and orthogonal to this plane. And that's how we're imaging activity at any time within the plane that's illuminated. Now, we have several special requirements that we place on this system based on the research we do. One is we want to minimize off-target sensory stimuli in the chamber. And we want to do that because we're presenting sensory stimuli of our own that we don't want to be interfered with. And the other is that we have a large amount of space to work with here. If we back up and take a view of this imaging objective, but also the part of the imaging column just above it, we see some of the customizations directed toward those goals. One is that we have a lot of space around this imaging area here. So if we need to bring in a screen, we need to bring in a speaker, we need to put in a microfluidics device or anything we might need in order to deliver sensory stimuli, we have space to do it. Another attribute of this system is that we always include a couple empty filter cubes here. This is so that we can pipe in, for instance, a laser here that we might use for optical trapping, or a spatial light modulator generated hologram. We can bounce that down there, for instance, to do a very targeted optogenetic manipulation. If we travel further up the light column, so everything you're seeing in this picture here is contained within this part of the microscope, and then there's a very long imaging column from there. And this gets at the key of how we minimize off-target sensory stimuli. We have the goal of doing volumetric imaging, but we don't want to move the animal through the plane in order to do volumetric imaging because we don't want to introduce, say, vestibular stimuli or water flow stimuli. We don't want the imaging objective pistoning up and down above the animal because we don't want to produce sound. We don't want to produce water flow there. So the solution employed by Idia Favreboul, one of the physicists in the lab, was to put an electrically tunable lens here in the imaging column. This is a lens that we can change the current across. It changes the shape of the lens. That then causes changes in the focal depth down here. We coordinate those changes in focal depth with the position of the light sheets that are produced by galvo mirrors, and that gives us the ability to do volumetric imaging without moving anything that's here immediately around the animal. So no movement of the chamber, no movement of the imaging objective. The overall outcome of that whole approach are images like these. So you can see here, this is an example where I'll just show you that volumetric imaging through depth starting at the dorsal side of the animal. This is a 6-day-old larva expressing GCAMP in the nuclei of all neurons. And you've just seen from the dorsal down to the ventral extent of this animal. So that's how we do volumetric imaging. Within that volume, we have a number of planes. Most typically, we have 50 different planes that we image at. And you can see an image here from one of those planes here in an animal that's responding to an acoustic stimuli. You can see activation here in some of the auditory centers of the brain. So we get this movie and we get 49 other movies simultaneously, and then we can reassemble that stack into a volume of the brain where we can resolve the activity of individual neurons. Usually we can run at 100 frames a second. Normally we do 50 planes at 2 volumetric hertz. If the experiment requires it, we can go to 25 planes at 4 hertz, or we can strike that balance between refresh rate and Z resolution wherever we want to set that balance, as long as it multiplies out to 100 frames per second. So that's the imaging approach. I'm going to go very quickly through what we then do with those data in order to infer what neurons across the brain are doing. So the first thing we need to do is find those neurons. For that, and as an example here, I'm just going to show you one structure. This is the optic tectum in one plane of one animal. This is going to be about 400 neurons, where we might normally get 20 to 30,000 neurons per animal. First, we stabilize the image. We perform a certain amount of pre-processing. We then perform segmentation. This just shows morphological segmentation. We now use the Suite 2P software from Carson Stringer at Genelia. Regardless of the approach used, the goal is to create regions of interest, ROIs, that correspond to individual neurons. And then we can extract activity in each ROI and each neuron through time during the experiment, and then describe what the activity of all those units are through time. In this case here, we're looking at the probability that neurons in the tectum will respond to a repeated visual stimulus. But the output always ends up being along these lines. The first look we get at it takes the form of a raster plot. And so you can see activity here in 400 neurons composing this one plane of this tectum in response to a series of sensory stimuli here. I'm not going to go into detail about what they are. But each neuron then has a particular relationship with different stimuli in this stimulus train. And this allows us to start to get a grasp on what kinds of cells are responding to stimuli, and perhaps how it is that details of those stimuli are encoded in the brain, or how information about those stimuli flows through the brain. The next step, and one I'm going to skip in a short talk like this one, is to start to make sense of what categories of cells there are, where they are in the brain, and how they might be communicating. That's a lecture of its own, so I'm going to skip it. I'm going to skip to what is essentially the bottom line of this process. In this case, I'm showing you raster plots from 5 different clusters. These are clusters that we produced using k-means clustering. They all belong to the same data set, but each cluster has its own characteristic response profile during a stimulus train. The stimulus train in this case is a series of visual stimuli, threatening Loom stimuli shown 10 times. You can see those here, a 5-minute break, another 10 exposures, another 5-minute break, and another 10 exposures. And the phenomenon that we're observing here is that of habituation, where most cells in the brain, and indeed the animal's behavior, drops off over time as this stimulus is shown repeatedly. The k-means clustering delivered clusters with these distinct response profiles, including those that habituated very strongly, stopped responding very quickly, moderately or slowly, maybe essentially unhabituating neurons. Neurons that are inhibited during the stimulus train, and neurons that are specifically coupled to the behavior of the animal. So these are the sorts of outputs we get when we look for activity patterns, look for characteristic responses across the brain. And then through a registration approach that I also won't talk about today, we can put them back into 3D coordinates within the brain. So these are the collective regions of interest from, I think it was 12 different animals, all registered to the same reference brain and put into a 3-dimensional context. They're color-coded to the response profiles they have. And what we get here is a visual sense of what the responses are in different parts of the brain. And this allows us, through a variety of computational approaches, to start to model how information flows through the brain and what the key points are, what the pivot points are for, say, sensory motor gating, for a decision to respond to this stimulus. So that's an overview of our method. There are lots of details left out, and if you're interested in those, please just contact me separately. But we've used this approach across a number of different sensory modalities over the last several years. These include water flow, where we designed a microfluidics device that permitted us to put water flow across the animal's body while it was under the SPIM, and to observe activity across the brain as the water was flowing across its body. It included vestibular processing, and this was the product of a very creative project from the same physicist, Idia Favre-Boule, who succeeded in putting forces on the vestibular organs, the ear stones within the brain, using an optical technique, an optical trap. So using a laser, she put forces on the otolith that actually tricked the animal into thinking it was moving when it was stationary. It was important to us that it remained stationary because that was necessary for our calcium imaging. So Idia wrote several papers using that approach. And the lab has also applied this method to both auditory and visual stimuli. The one I'm going to talk about today, because it's just recently come out and I haven't really talked to this community very much about it, is a description of the auditory system and sensory processing within the auditory system of larval zebrafish. Now, historically, the auditory system of larval zebrafish has been viewed as very rudimentary, so only responsive to low frequency stimuli and without particular tuning to stimulus properties like those with sharp onsets or those with particular specific frequencies. An excellent PhD student in the lab who's just graduated, Rebecca Poulsen, whose background was in acoustics specifically and being a DJ, developed a system that overcame a long-standing problem in doing this kind of imaging using acoustic stimuli. So for aquatic animals, you have to give a lot of thought to how you're applying acoustic stimuli. In a large body of water with an underwater speaker, the animal's likely to get about what you mean to deliver to it. But if you've got an air speaker and an animal in water, that air-water interface really interferes with the delivery of the stimulus. A chamber of this size, which is necessary for calcium imaging, is too big for an underwater speaker, and the acoustics inside a chamber like this are too chaotic anyway. The solution that Rebecca came up with was to use a tiny speaker, this is about 9 millimeters in diameter, affix it directly to the cover slip slide wall of the chamber, and effectively turn that cover slip into a speaker that delivers those stimuli directly to the animal. And so in doing this, she was able to deliver with much better fidelity exactly the acoustic stimuli that she meant to. So she had some fun with this. She played Zebrafish Larvae, You Can't Touch This by MC Hammer. And I won't show you the video or play the music, but she tweeted about this in what is no doubt going to be the high watermark of my lab's fame. The good Reverend Hammer actually retweeted this himself. This was a zebrafish larva here responding to his song. But Rebecca also put this approach to more scientific use and designed really quite a complex acoustic stimulus train that she could play to these animals. And that's represented in this diagram here. Here, grayscale represents white noise, so just a static sound with lots of different frequencies included. And individual colors represent individual pure tone frequencies. And so she created a stimulus train where there were, for instance, white noise crescendos. This here is the amplitude of the sound. Loud pure tones, white noise, discrete white noise stimuli of various amplitudes. Those were each shown 3 times in various orders. And then this is a frequency sweep, so like a siren sound from low to high frequency or high to low frequency. This stimulus train provided all sorts of opportunities for the auditory system to show what it was capable of. And so she then did a study revealing what in fact can happen inside the larval zebrafish auditory system. To make a long story short, for a short talk today, she found all sorts of things, including all sorts of things that hadn't previously been described in aquatic larvae. She found, for instance, broadly responsive, so not particularly selective, neurons that nonetheless had some preference for complex wavelengths, that is to say, white noise over pure tones. And you can see, for instance, here, strong response to a white noise crescendo, responses here to a white noise stimulus that are stronger than the corresponding responses, white noise stimulus corresponding responses to pure tones, even though they're at the same volume. The volume to this stimulus, the volume of this stimulus here is the same as the volumes of any of these stimuli here. She found broadly responsive cells that had the reciprocal preference for simple wavelengths, pure tones, where the responses to several pure tones were stronger than to those responses that she saw to white noise. And then she saw some neurons that were virtually selective, virtually exclusive to pure tones. You can see a range of pure tone responses here, where the responses, so this stimulus here is as loud as any of these stimuli here, and yet something about this white noise stimulus is not being picked up by, not being responded to by these cells. So she saw those sorts of responses. And because we can then register things and put them back in the context of the brain, she could identify the regions of the brain in which these cells were located. And in this talk, I'm not going to go into the details of how these localizations speak to the likely flow of information, but that's part of where this project next went. There were still more specific responses that we found very interesting and that were novel. Responses, for instance, to short waveforms, so those stimuli with sharp onsets. So you can see here are white noise stimuli from loud to quiet. Here's a white noise crescendo that ends at the same amplitude here, the same volume as this stimulus here. And you can see the ratio of these responses here. So these are cells that are especially responsive to tones that have a sharp onset rather than a gradual onset. She saw the reciprocal, cells that were responsible specifically to crescendos and virtually ignored those stimuli, even equally strong stimuli with sharp onsets, and those responsive to frequency sweeps. So again, these are these siren-like stimuli. And so a lot of what she did next was make inferences about where in the brain these responses were taking place and how then information might be arising, how novel features, emergent features of the auditory system might be produced. So that covers auditory processing in general. She was also interested in those cells that I talked about before that were specific to pure tones and interested in judging whether the animals might actually have frequency discrimination, the ability to distinguish high from low frequency stimuli, which hadn't been shown in larval zebrafish before. To do that, she took those pure tone responsive cells and she did a regression analysis and assigned each cell to the frequency that it preferred. And then she could map that back onto the brain. And what she found is that they were pretty well intermingled. So there's nothing like what you see in mammals, where you see tonotopy and where low frequencies are encoded here and high frequencies are encoded here. And there's a continuum among those in space versus frequency. But she did find brain regions that specialized in responding to low frequencies or intermediate frequencies or high frequencies. Again, you can go back to the types of naturalistic stimuli these animals might be experiencing and try to make inferences about what the relationships are between the natures of those stimuli, the responses in the brain, and the adaptive behaviors the larva would have in response to those stimuli. So I've just provided a very brief and a very shallow overview of Rebecca's work on the auditory system. This is all in wild-type animals, and this provides a baseline from which we can now depart on another thing that the lab does. So we also do modeling of human conditions, specifically with sensory processing and specifically with brain regions, subcortical brain regions that are fairly well conserved between fish and mammals, including humans. So I'm going to finish with a very quick example of that. In order to trial this whole approach, we took a very well-established and a syndromic, which is to say a single locus human disorder, Fragile X syndrome. And we took a zebrafish mutant with a mutation in the conserved gene, the FMR1 gene in zebrafish. And this started with a very broad then sensory profiling of mutants versus their wild-type siblings using both visual and auditory stimuli. And to make a long story short, when Lena Constantin, a postdoc in the lab at the time, looked at responses to visual stimuli, she didn't see any marked differences between the controls and their mutant siblings. But when she looked at auditory stimuli, and this was with a preliminary, fairly shallow stimulus train of acoustic stimuli, she saw more cells responding in the mutants and responses over a greater region of the brain. So then with a second data set, very much along the lines of what we've already shown for Rebecca, and indeed Rebecca collected these data, presented a wide range of different auditory stimuli and looked at the responses of the FMR1 animals versus their wild-type counterparts. And then started performing various quantifications of these responses, including quantifying responses in particular brain regions of interest. And by doing this, she could look, for instance, at a structure. This is the torus semicircularis. This is the second location within the central nervous system that receives this auditory information. And she found that as judged by the proportion of the cells in that brain structure that were responsive to auditory stimuli, in terms of the strengths of the responses of those cells, she didn't really see any differences between wild types and mutants insofar as the networks are different. It's probably not because they're different in this brain region. Compare that to what she found in the thalamus, where she found that a much higher proportion, each dot here is an individual animal, a much higher proportion of thalamic neurons were responsive to auditory stimuli in FMR1 mutants than were in wild types. And at many different amplitudes, these are weak stimuli, pardon me, these are weak stimuli up to strong stimuli. At several different amplitudes, she significantly greater responses, stronger responses in the wild types, in addition to there being more responsive neurons. Now, one way to quantify this and to get a sense of what might be happening across the brain-wide network is to employ graph theory. And so graph theory is one of several approaches, several mathematical approaches we're applying to try to make sense of these data. And it's the form I'm going to present today is a very rudimentary form of graph theory that just allows us to get a snapshot of the brain as a whole. So the way this works is instead of looking at tens or hundreds of thousands of neurons at a time, we need to downsample, we need to coarse grain. And so what Lena did in order to do this, again, Lena Constantin, what she did was she created nodes that each represented a large number of ROIs. And for a given node, its position and its response profile reflected the usually hundreds or thousands of ROIs that were composing the average signal for that node. This again is a means of just coarse graining the brain-wide activity to a scale where we can perform these sorts of analyses. So there's a wild type animal, and these are the nodes that you could form in wild type. A first step then in graph theory is to look at correlations across these nodes and to fill in edges, draw lines between nodes if their correlations exceed a certain threshold. And when we did this in wild type, what we found is that at quiet stimuli here to gradually stronger stimuli, you start with very little in the way of connected edges because there's little correlation across the network, up to very strong stimuli here where basically the whole network is interconnected. If we look at those same volumes, but representing data from the fMR1 data, what we find is that actually the structure of the network is not so different. The nodes that are connected are in many cases the same nodes, but those connections take place, those correlations are higher at lower amplitudes, at quieter volumes. So this speaks to a network that's overall livelier, overall more sensitive, and in ethologically important ranges, probably a little bit overactive. This very much reflects the mammalian literature, reflects the existing knowledge of auditory hypersensitivity in mammals, auditory hypersensitivity in humans. And so this then gives us a departure point, a basis on which to model what might actually be happening anatomically or physiologically in these animals. We can view these data either with anatomical context, so where the nodes are positioned spatially, or we can take a more functional view. These are all the same nodes, but now they're separated by brain region around the outside of the ring. And we can look again at how these networks are more active at lower frequencies in the fMR1s than the wild types. So that provides a very brief overview of our approaches, how we can use it to describe brain-wide processing of a specific modality, audition, in wild-type animals, and then in turn use that as a departure point for doing phenotyping in models of human disorders. All of it's been very shallow, all of it's been very high level today because it's a quick talk. If you're interested in any of the details, any of the caveats, anything to do with the talk, I'll be happy to take your questions now. And you're always welcome to contact me after the fact and discuss these things. I'd just like to wrap up by again thanking the organizers and thanking you, thanking the members of my lab, collaborators with our work, and funding sources. So thank you very much, and I'll take your questions now.